Good morning, ladies and gentlemen. Welcome back. Our next speaker champions interdisciplinary research, and in recent years, his research has expanded into the area of high-frequency medical devices. For example, the non-invasive blood glucose sensor technology presented here, including work on the non-invasive blood glucose monitor we're about to hear about. So please welcome Professor Adrian Porch.
Thank you, everyone. Hope you can hear me. If not, shout and I'll try and talk louder. Thank the organisers, Ian and others, for asking me to give the talk at this meeting. So my name's Adrian Porch. I'm a professor at Cardiff University. I'm a physicist, I'm not a medic, and I work in engineering. So we basically do things with high-frequency circuits, basically. And this is a problem I've been working on for about 15 years now. It's a bit of a holy grail, obviously, in terms of blood glucose monitoring. Can you have a blood glucose sensor that doesn't require access to blood, you know? So it's non-invasive. It obviously needs blood in the body, but you don't need to prick your fingers, etc. So it's basically that. So I'll tell you a little bit about the technology and trends here relating to non-invasive blood glucose sensing, because it's been a hot topic for, you know, at least a couple of decades now. And tell you a little bit about what we've been doing more recently in terms of work with GlucoRx. And we've got Neelish Nathwani, who's CEO with GlucoRx. Oh, there he is, yeah, who's here also today. So I'll tell you a bit about the underlying technology. Obviously, I can't tell you absolutely everything, but I'll try and explain the basic concepts. And we put our earlier generation devices through clinical studies. So I'll show you some of that clinical data and then mention some of the future plans for this technology. Now, diabetes itself, probably don't need to tell you, how major healthcare issue it is worldwide now, especially in developing countries. And it was estimated that 12% of global expenditure is currently, put to diabetes and conditions related to diabetes. So we need an affordable solution here for the management of the condition, just to make it more easy. And obviously there are subsets of the population, for example, young children, who you don't want to be pricking their fingers or trying to do anything invasive with them in terms of trying to monitor their blood glucose levels. So the current state of the art regarding continuous glucose monitors, CGMs, they're chemically based sensors. I haven't got a laser pointer here, I don't think, but you can see here there's a sensor that basically has a needle on it that sits subcutaneously, and then it sits in interstitial fluid. So it measures the glucose level there, so it doesn't actually go into blood. So there's a delay, which could be an issue, and that delay could be up to 15 minutes or something like that. And that's a pretty big issue for people, for example, with type 1 diabetes. The delay, depending on the other types of or minimally invasive methods, depending on what you're probing, can be as long as 45 minutes. And what we've got here, a sort of overview of some of the partially invasive type sensors. Some of these involve microneedles, so they may not involve pain, but they certainly involve the exchange of bodily fluids. So we've got things like the Freestyle Libre there, and the Dexcom type CGMs. And then on the right, you can see some of the attempts that have been made to develop non-invasive CGMs. Now, Google Glass was quite popular, blew up about 5 years ago or so, but then that went away. And then there are other companies were set up with apparently non-invasive blood glucose sensors based on electromagnetic technologies, and more about that in a minute. But they've come and gone. So obviously this is quite a difficult problem, and it's by far the most difficult problem I've ever had to deal with in my research career, actually. So, but we're getting there. So we're looking at this, you see, you've got the various types of fluids that you can access, and you really want to get at the blood. You don't want to be working with sweat, breath, urine, simply because of the delay involved, So how do we get at that blood? Well, these are the electromagnetic type sensors that have been developed over the last couple of decades. And some of them are watch-like, so This one works at RF frequencies, radio frequencies. And what you see there, this is published data, you see trends which track blood glucose. I think these were done in comparison with some blood prick tests at the same time. This is the GlucoRx where, again, the actual model of this sensor is the one that you can see there, which looks like a 3D printed part to me. The actual device is lab-based, so somebody sits there next to a big box of lab equipment, you see. So the reality is far removed from, what's being promoted. And this is going up to terahertz frequencies now, more towards infrared type frequencies. You see a poor rat there who has a terahertz waveguide. You can see that the rat's ear is being interrogated with some terahertz signals. But again, there is a signature of blood glucose in the dielectric properties that are measured in all of these different types of sensors, you see. So this is our pathway. Basically, I've been working with this sort of stuff for all my research career now, which is 30-odd years. And I put things in high-frequency fields and then see how they respond. So obviously, the thing we're talking about here is the body and blood specifically. So since 2008, Cardiff, we've been developing non-invasive methods for studying blood glucose using electromagnetic means. The work was initially sponsored by Cardiff University, but then we got a fairly major Wellcome Trust Translation Award, which allowed us then to develop our first prototype devices. So I name check Wellcome here frequently because they really have been the instrumental factor really to getting us where we are now, and of course in partnership now with Nilesh and GlucoRx. And we've been working with GlucoRx over the last 3 or 4 years to try and make this a commercially viable prospect. This is the device that we're actually moving towards now, and I'll say something more about this yet. You can't get this at the moment, but this is the Bioxensor, which we say is our 4th generation device. I'm going to tell you mostly about generations 1, 2, and 3 in terms of how we got here, and I'll come back to this right at the end. I'll leave some time for questions, by the way, at the end as well, so hopefully we can address a few of your questions then. A little bit of techie stuff, because I'm a physicist. You recognize that, I hope, as an electromagnetic wave. There's a red bit, which is the electric field, and then a necessary blue bit, which is the magnetic field that makes that wave. What we're interested in is the interaction of the red bit, which is the electric field, with, in this case, the body, and specifically blood, and any changes that could happen in blood. So we use RF frequencies. There's a very good reason for using RF. First of all, they're safe. They're non-ionizing. If you go up in frequency, things do become a little bit more dangerous, actually. You eventually get to X-rays. We won't go there. But people are worried about 5G and 6G, and that's just doubling the frequency, say. So we keep down at RF-type frequencies. It's non-ionizing, but more importantly, we can penetrate the body by a few centimeters. So it's not a surface measurement. You are getting at blood vessels. You all know that you can cook your baked potato in a microwave oven, for example, right? That's a few centimeters in diameter. And we're using here very low power levels, way below the levels of a mobile phone. So it is electronically safe. Again, a little bit of techie stuff. This is what we're trying to target here. What we've got here is a plot of what we say is the dielectric spectrum of materials. So this goes all the way from radio frequencies through to infrared, through to visible light and UV. It's a log scale. And you notice there that there's all sorts of blips going on. Those are different sort of mechanisms that we can target with that red bit of the electric field. Now, what we're talking about are RF frequencies, and they actually tend to cause things like water molecules to rotate, all right? It's that rotation and the friction because of that rotation that gives rise to heating of microwaves in domestic cooking, all right? What we're doing by targeting that particular rotation is to look at its disruption, the disruption of the hydrogen bonding network by adding something to water. In this case, it's the presence of glucose. But it's a trace, it's a needle in a haystack, this. It's not something that's easy to extract. So we've got to get the electronics right. So we're looking at rotations of water molecules and the disruption of that from other species, such as glucose. Very good question is, how specific is it to glucose? I'll come back to that in a bit, because it isn't specific to glucose, but that's why we have to have other sensing technologies on board as well, and that's really important. So the RF method is going to give us some information, the primary information, but it's not going to tell us the whole story. So we need a multi-sensor approach here. A little demo then. It's a bit too early to be drinking wine, so I haven't got any wine glasses here. I've got a bell, right? I'll try and illustrate the sort of principle of the measurement here. That's called a high Q resonator, okay? Basically, it's a ping, isn't it? If you look at the oscillation of the bell, the oscillation carries on for quite a few seconds if you listen to it. And it'll keep going, all right? It'll dampen down eventually. I haven't got enough hands for this, sorry, but if I thought I'd have a wearable mic. If I touch the bell and ping it, then listen to the difference. Technically, that's called a clunk. And if you look at a clunk, the oscillation decays much more rapidly. Now, what we're doing is we're not using acoustic resonators, we're using electromagnetic radio frequency resonators, which are quite small, okay? They can be packaged so that you can wear the things. And we don't look at things in this so-called time domain. We do something called a Fourier transform of this and look at things in the frequency domain. So if you look at the ping in the frequency domain, you have this really sharp resonance. But if you damp it, the clunk looks like that blue trace. So two things happen. One is you get a shift downwards in resonant frequency, and you can hear that with the bell, can't you? Because it rings at a much lower frequency, or not much lower, lower frequency. The other thing, though, you notice the width, the spectral width or the bandwidth increases as well. So there's two bits of information for us there that tell us about the thing that is causing the damping. So it could be the wine in the wine glass, but it could be the body when you attach one of these RF resonant sensors. And then we can track these properties, you see, and then try and track them as a function of blood glucose changing within the body. And that's fundamentally what we do. So we do this all the time in my labs. You know, we have boxes like this that you pay Keysight, you know, £50,000 for, and you can do these sorts of measurements. You notice there's a nice ping going on there, okay, on the bench. What we've done with Wellcome over a period of about 6 or 7 years or more is to scale that down into a wearable sensor. So this is Hongjae Che, who's one of my colleagues at Cardiff, who's done a lot of the legwork on this, actually. And that's the first, that's actually the Gen 3 wearable sensor that we developed with Wellcome funding through the Translation Award. So it's basically taking on board all that onboard electronics on the box and putting them onto this wearable device. So it's got a rechargeable battery in there, and you notice it's communicating with not a mobile phone, but with a laptop computer. But it's fairly simple in terms of the system to imagine that working on an app. Okay, which of course where we're going with the next generation device. So we've gone, these are the 3 generations that we took through to with the Wellcome funding. The first ones were wired, you know, so they did involve somebody sitting next to a box of electronics or moving around, you know, as you do in a hospital or clinical ward with one of these trolleys, you know, with all the electronics on it. And then we moved to a wireless solution. And we've done the modeling of this, so you know, we're not doing this in the dark because we are experts at material interaction at high frequencies. So what we've got here is models, for example, of the skin And if you've got good eyesight, you can see here, this is a 3D electromagnetic simulation of the radio waves from our sensor penetrating through quite a few millimeters of skin, getting to fairly deep blood vessels, which is what we want. We tend to wear the sensors on the arm or the upper torso because we want to try and minimize movement. And this is basically how we generate a signal. You notice here, I don't have a pointer, sorry, but you notice the ping type measurement. We basically do a curve fit on that, extract the resonator parameters of frequency and bandwidth, and then we correlate those with known values of blood glucose. So what we've done is we've run 4 clinical studies now. I'll say something about that in a minute. And in those clinical studies, the subject is fitted with a cannula, so we have access to venous blood as and when we want it. So the subject goes through an oral glucose tolerance test, which some of you may know of, but that involves just drinking a solution of known concentration of glucose, and then subsequently your glucose level will go up and then come back down or stay there, depending on your condition. So we know gold standard what the blood glucose is at any instant in time, because we use something called the Yellow Springs instrument blood glucose analyzer, which I think is the gold standard for these types of measurements. So we thank Steve Luzio, who's one of our colleagues, was at Cardiff, but now is a professor at Swansea. They've got an excellent clinical trials unit down there. So he does the clinical. I'm not really qualified to talk about a lot of the information on this slide, but it's a typical sort of study. This was a 24-subject study. The most recent one we did was 50 subjects, which I'll show you the results for in a minute. So some of these are non-diabetic, some have type 1, and most of them have type 2 diabetes. And this is what we measure. So we measure the blue trace here, which is the RF measurement of blood glucose. The red one is what we get from the Yellow Springs instrument. I'm being fairly selective with this data. Sometimes it's not as good as this, and I have to be fairly honest with you here. But there's definitely a signal there. Other people are measuring it, and there's a bit of a race at the moment as to who will get to the finishing line first in terms of a commercial device. If you put these on the conventional grids that are used for correlating blood glucose readings for such sensors, you've got something called a Clark error grid, where you're basically plotting your measurement vertically of glucose concentration against that of the gold standard. And of course, if you get the axes right, that should be a straight line through the origin, a 45-degree angle, And we sort of get that, the scatter in that data. This was a 50-subject trial, but there's nothing there to cause any major concern. You notice we get good correlation over a very wide range of blood glucose values. And it doesn't depend really on whether the person is a type 1 or type 2 diabetic. We've got more limited data for type 1s than type 2s, but you can see that they're all in the mix really. This is quantified by a MARD value, mean absolute relative difference, and it's about 10.4% here for those devices. But this is done in a clinical setting, and we do tell people to try and keep still. We can tell if they popped out for a fag and things like that, in terms of things like temperature difference, nonetheless. And they do this without telling us, of course, but we know. We can tell from the data. It's a pretty good motion sensor, this, as well. So this is where we're trying to head, anyway, with Nilesh on board, is towards the 4th generation sensor, which is our Bioxensor. But in there, we have to have other types of sensing technologies. Ideally, other types of other parts of the electromagnetic spectrum, because the RF is going to give us really good information, but it's probably not going to be good enough on its own. So this is wearable. It can probably be made smaller than this, as technology improves, things like battery technology, for example. The current dimensions, 42 millimeters by 7 millimeters, say 42 millimeter diameter, 7 millimeters thick, weight of about 14 grams. So it's not as small as the CGM sensors that you see, but it's still, very convenient, comfortable device, I think, when it's on the arm. But in there, we've also got things like near-infrared, because I showed you earlier the electromagnetic spectrum, because you get different types of behaviors, different parts of the spectrum. So if we want to try and remove artifacts and interference, we can't just look with black and white vision. We need color vision here, which is why we do measurements over a broad range of frequencies and wavelengths. We've got BioZ, which works at very low frequencies, so that takes out things like electrolytics, for example, because obviously there's not just glucose that's changing in blood, there's all sorts of other things happening as well. We need accelerometers in there because if you move, there's a jolt in the measurements, so we have to account for that. And this is where AI is going to play a role, because I'm not an AI person. I deal with electromagnetic interactions with materials. We generate a signal, which is pretty good, actually. But put this in the hands of an AI specialist. They'll be able to fuse this data, train it, and who knows, what sort of marked value we'll end up with. We patented this, so a patent has pretty much gone worldwide now on the basic sensing technology. And this is the team that we've assembled, thanks to Nilesh, at Cardiff University and at GlucoRx mostly. And again, I give a very strong thank you to Nilesh for supporting this work. And Nilesh, I think, is very happy to take questions off anybody relating to the commercialization angles of this device. I'm obviously one of the tech heads, really. But I've come in about 8 minutes early there because I know we're on a tight schedule. And I'm sure that some of you will have some questions as well. So I'll stop there and open the floor to questions. Oh. I can hear you. I can probably repeat. I'll repeat your question if you want. Okay. What's on here?
Great results. Good to see, clearly you've made great progress and you'll be on the 5th gen before we know it. Clearly there's a good approximation or a good proxy between the RF readings and the venous readings. How do you validate that? Is there some standard that you're working to? What proxies would you use for that?
The only thing we can do there is to compare, our results with known blood glucose values. So what we do is through the clinical studies are obviously MHRA approved. So within the study protocol, we say we take blood every, say, 10 minutes after the OGTT, or glucose tolerance test. And all we can lean on, really, is the results that we get from that blood glucose analyzer measurement. So we take 3 measurements of venous blood. So, we're not just doing it based on one measurement, and then we do our statistics based on that. And then we're correlating our sensor with that of the gold standard. But apart from that, we haven't got any other way of measuring it. So we are a bit of a slave to that. Yeah, we'll have a chat later, yeah.
Firstly, really impressive talk. Thank you very much.
Thank you.
And I'm a diabetic sufferer, type 2.
Okay.
So it's a brilliant news for many years. And of course, I will talk about the price afterwards. But anyway, and you mentioned something very interesting, actually, about the motion detection.
Yeah.
And I feel that there must be a way of algorithm you can actually correcting that motion.
Yeah.
And then there will be some more work can be done, like a continuous monitoring of the patient at any kind of activity.
Well, that's right. It's not just a blood glucose monitor, you see.
Yeah.
And that's the primary signal that we want to get. But there are lower hanging fruit than this. I've already said it's a needle in a haystack, to get the blood glucose out of there.
Yeah.
But in terms of vital signs monitoring, we've already have, a very useful sensor here.
Yeah.
And I know there was a talk earlier, for example, on stroke and early intervention after stroke. You know, this is another example of a sensor which, you know, people don't want cameras on them, you know. They don't want to be, because, you know, they don't want to be, especially in things like bathrooms, you know, where people are vulnerable. So you need a sensor that will just give information relating to the state of the individual. And glucose is just one thing.
My second question is more about the data.
Uh-huh.
You must have collected quite a lot of data.
Yeah, we got lots
If
and lots.
this is a wearable device, we can actually turn it on 24 hours a day. And
that's right, yeah, yeah.
So do you actually have these data collected and made publicly accessible by researchers?
The clinical data is all accessible. We haven't published it all yet, actually, so it will be in the public domain because it's publicly funded research. The interesting thing is everyone shows you clinical data. These are done in clinical trials units. The duration might be 2 hours or something. They don't put it on a person and then send them away and come back in a month and then see what happens. So we're sort of in that stage now, you know, of the real life scenarios where we do have to contend with, dare I say, environmental issues, you know, because there's temperature. We have to measure temperature because temperature messes everything up, you know. It's the bane of our lives, really, when we're trying to measure properties of things. So, you know, clinical data, take it with a pinch of salt. It works, and then we have to make sure it works, you know, in a real-life scenario. Oh, yeah.
Thank you so much for delivering your speech. I have a couple of questions. One is regarding, you said, because there is a 15 minutes delay for the freestyle Libre, let's say.
Yeah.
Right? And what's the delay in your device
Yeah,
at the
good
moment?
question. I didn't actually state it explicitly, but we don't see any delay. We see an instantaneous change because what we are averaging interstitial fluid and blood, you know, so there's probably you know, a mix of the two, but we see the trigger, you know, the rate of change is exactly the same trigger of glucose in blood as what we see in the venous blood. So we don't actually see a delay, which is one of the important features, you know, of having a non-invasive sensor that looks at blood.
Sure. Another question, because it says it's a wearable device, but it seems like it's like kind of freestyle Libre place where it's going. So
yeah.
You have to wear it for a few days, or it's just kind of a sticky bit underneath that?
You can stick it on. The interesting thing with our sensor is it's non-chemical, so there's nothing actually to wear out in it. It's battery operated, so you know you have to recharge the battery, but there's nothing chemical there, you know. So with the Freestyle Libre, you do have to take that sensor out at some point. Our sensor just keeps sitting on the skin, you see. So there's nothing. It's purely electronic based. So it can go in any location, but some locations are better than others in terms of trying to remove some of the artifacts, you know, especially if you're moving around, for example. So, you know, we'd like to put it somewhere where it's fairly discreet and you know, it's not going to be subject to too much motion artifact. So top of the arm is a good one.
All right, just last question.
Yeah.
In your presentation, you mentioned that the signal go further beyond the skin, right? But you are measuring the glucose in the blood, in the real blood. How do you know that when you send the signal, it is targeting the blood only, not the combination of the dielectric properties where it's interfacing?
We don't actually. The thing is, the proof of the pudding is the eating here in terms of what we correlate it with, you see. So we're correlating our measurements with that of a blood glucose analyzer, and we're getting that sort of level of correlation. So I think we are measuring, you know, the dielectric property of anything that we put underneath our sensor.
Yeah.
But there's certainly a large blood component there that we are picking out, and that is being traced in our data. But I think, you know, absolutely we're measuring interstitial fluid as well.
Yes.
You know, because everything under the sensor, it's not going to pick out blood, you know.
Sure.
It's just going to measure everything that it sees.
Sorry, just last question. You mentioned about the ping and chunk, right? The dielectric property.
You're from Avon Technologies,
yes, I
yeah?
do. But my background is the non-invasive.
Yeah.
So imagine, so you send a signal in the body.
Yeah.
And
we don't send a signal in the body.
No, I know, I understand that. You're looking at the transmission coefficients. So imagine if there's a kid there, the dielectric properties for the kid has the higher concentration of water there
Yeah.
as compared to the adult man,
Yeah.
right? And let's say the kid is diabetic and the adult is also diabetic. So when you look at the same device,
Yeah.
it's possible that because of the dielectric properties of kid, you can absorb more energy because you're looking at it.
Yeah.
You're going to see the different chunk as compared to the adult. So how do you differentiate between?
Well, that's why we use we just don't use RF,
Okay.
okay?
Mm-hmm.
We're not pinning all our hopes on RF,
All right. Okay.
okay?
Okay. Thank you.
Thanks very much, Adrian. I think we
6
have
seconds.
well, 6 seconds left.
I
but
can answer questions outside.
yeah, exactly. I know this lady here in particular wants you to go and talk to her afterwards, so I'll point you out. Anyway, thank you very much for attending Adrian's talk. Next, I'm going to leave you in the capable hands of Chris Rowe from Innovate UK. So, thank you very much.
Thank you.